Hello and a warm welcome everyone to today's webinar on ligand assays, a versatile and acceptable approach for clinical safety and immunogenicity assessment. I'm your host for today and my name is Bhavika Maheshwari. I represent Veeda Lifesciences. As a global integrated and tech-driven CRO, we are offering a comprehensive portfolio of preclinical, clinical, and bioanalytical services to support our global clientele in their innovative, biosimilar, and generic drug development programs. With an extensive global network of key opinion leaders and clinical sites situated across 26 geographies, today we have become a trusted drug development partner delivering results on a global scale. Together, we are catering to industries like pharmaceuticals, biopharmaceuticals, agrochemicals, and industrial chemicals, herbals, nutraceuticals, and medical devices industries. We are extremely driven by a focus and a continuous mantra in achieving excellence in everything we do. We yearn to continuously innovate and deliver cutting edge solutions to all our clients. That was a little bit about our CRO. As a facilitator for today, I would like to introduce you all to a couple of pointers before we begin the session. This session will have a presentation in the beginning and this will be followed by a Q&A session. We have enabled ask a question feature which is there on your screen. You can drop all your questions related to this webinar over there. Moving forward, let me quickly introduce our speaker for today, Dr. Swaroop Sarkar. Dr. Swaroop Sarkar holds a PhD in biotechnology from Bharatiya University, Coimbatore. He brings over a decade of experience in bioanalytical assay development for biologics. Currently, he is serving as an analytical project manager at Veeda Biopharma in Bangalore. He is leading the clinical bioanalysis for GCLP compliance studies, and he specializes in PKPD, ADA, NAB immunoassays. His extensive portfolio works in biosimilars, peptides, uh, ADCs, SCFES, and bispecifics. His past tenures include Zydus Research Center, Biological Limited, Apollo Health and Research Foundation, and Indian Veterinary Research Institute. So, without any further delay, I'd like to virtually call upon Dr. Swaroop Sakar to begin the session.
Good evening, everyone. As Bhavika has already introduced me, so I will not go into the introduction again, and I will directly jump into the topic of today's webinar, that is ligand binding assays, a versatile and acceptable approach for clinical safety and immunogenicity assessment. Ligand binding assays has become a critical tool in clinical bioanalysis of large molecules. They have become an important part of bioanalysis and they are playing a critical role in the bioanalysis of large molecules. They are widely acceptable and versatile in nature because they are reliable and reproducible. Here in this presentation, the key intakes will be the types of ligand binding assays, the importance of ligand binding assays in clinical research, and the challenges that are associated with the ligand binding assays, and how they are impacting the clinical safety and efficacy of the product. Starting with the definition of ligand binding assay, is ligand binding assay is a biochemical reaction or a biochemical test which shows the reaction between a ligand and a target, how efficiently it is binding to the target, and after binding to the target, it forms a ligand receptor complex, which is in turn produces a response which is measurable, and this response is directly proportional to the concentration of the ligand that is present in the sample. These ligand binding assays are versatile and can be adapted to various applications like PK, TK, immunogenicity, and biomarker analysis that are used in the clinical safety and efficacy assessment. This provides an insight into the mechanism of action of drugs also, how the drug is absorbed into the body and how it is distributed to the different parts of the body, how it is eliminated and excreted from the body. Ligand binding assays are used to measure the binding affinity between the ligand and the target. How strongly this ligand is binding to the target. Usually the binding affinity is measured in terms of dissociation constant, that is Kd. And this Kd is inversely proportional to the binding affinity. The higher the Kd value, the lower is the binding affinity. So this becomes a important parameter or criteria for the selection of positive controls in LBAs. So whenever we are designing an assay, we have to select an positive control which is having a lower Kd so that it binds to the ligand tightly and gives a higher sensitivity or specificity reactions. This also helps in the investigation of mechanism of drug action, how the drug works, how it is absorbed, metabolized, and eliminated from the body. This is used for the quantification and estimation of concentration for the PK as well as PD parameters, thus supporting the PK and PD studies in drug development. Coming to the different types of ligand binding assays, there are several different types of ligand binding assays like radioimmunoassays, RIA, enzyme-linked immunosorbent assay, ELISA, SPR, surface plasmon resonance, fluorescence polarization, FP, and alpha LISA. These are few of the types of ELISAs that are used in the ligand binding assays and have an importance in clinical bioanalysis. So coming to radioimmune assay or RIA, it's a type of quantitative assay which is used for the quantification of antigen or haptens. So how this RIA works? RIA is a type of competitive assay where the radio-labeled antigen competes with the unlabeled antigen that is present in the sample. When radio-labeled antigen is present or the antigen is absent in the sample, the radio-labeled antigen goes and binds to the antibody and gives a highest response. So when the antigen is present in the sample, this antigen is more specific than the radio-labeled antigen. So it tries to displace the radio-labeled antigen from the antigen antibody complex and thus reducing the intensity of the color developed. Hence, this is indirectly proportional to the amount of antigen that is present in the sample. The lower the antigen present, the more is the signal of the assay. Next, coming to SPR. SPR is a surface plasmon resonance reaction, which helps in the study of binding kinetics and behavioral or functional properties of biomolecules. SPR is based on the RI index, or that is refractive index of the molecule or light. When antigen binds to antibody, it tends to change the refractive index of the light. So it depends on the binding of antigen to the antibody, as SPR is a chip-based technology where the antibodies are bound to the metallic surface of the chip. And when antigen binds to these antibodies, there is a change in the refractive index of the light. The light tends to bend slightly towards the right. So this bending or the refractive index is depending on the binding of the antigen to the antibody. And based on the specificity and affinity of the antibody, it produces the association and the dissociation curves. Alpha LISA. This is a luminescence-based technology, which is a great tool for studying the protein-protein interaction. Here in alpha LISA, it's two different types of beads are used. Two different types of beads are used. One is the donor bead and the other is acceptor bead. The donor bead is conjugated with streptavidin and are designed to bind with the biotinylated antibody. And the acceptor beads are conjugated with the antibodies that can bind with the same analyte that is present in the sample. So these two binds together and brings the beads, which is a donor bead and acceptor beads, to the closer proximity. Based on the closer proximity, the donor beads get excited and it gives or transfer the oxygen molecule to the acceptor one, which in turn produces the luminescence reaction. Coming to fluorescence polarization reaction, ELISA. Fluorescence polarization ELISA is used to provide the information on molecular orientation as well as the mobility and the process of that modulates them. FP actually measures the changes in the rotation of fluorescently labeled ligand, which is bound to the target. So it is a type of competitive ELISA only where the ligand is fluorescently labeled with a ligand, and in the absence of antigen in the sample, this fluorescently labeled antigen goes and binds to the antibody, and the rotation or the ligand rotation is slower. It causes an increase in the polarization. And in the presence of sample, this ligand is, which is fluorescently labeled ligand is displaced, and the antigen binds to the antibody, thus decreasing the polarization of the assay. ELISA. There are several different types of ELISA which can be used for quantitative as well as qualitative purpose. So for qualitative assays, we can detect different types of antigens or antibodies. And in quantitative assays, we can quantify the amount of antigen or antibody present in the samples. ELISA is a simple enzyme-based reaction which converts the substrate into a colored product, and the intensity of the color that is developed is directly proportional to the amount of antigen that is present in the sample. There are different types of ELISAs that are available to and are used in the clinical bioanalysis techniques. Direct ELISA, indirect ELISA, sandwich ELISA, bridging ELISA, competitive ELISA, and MSD. Direct ELISA, coming to direct ELISA, it's a direct quantification of antigen that is present in the sample. It's a one-step ELISA where the detection system directly binds to the antigen that is coated onto the surface of the ELISA plate. Indirect ELISA is a two-step ELISA, which is used for the quantification of the antibody that is present in the samples. It is the antibody that is present in the sample binds to the target that is coated onto the surface of ELISA plate, and this bound antibody is detected by a conjugated anti-drug antibody. Sandwich ELISA is also a type of bridging ELISA. Sandwich ELISA is a type of quantitative ELISA which is used for the quantification of antigen or antibody that is present in the biological samples. Usually it uses two different types of antibodies. One is primary antibody and the secondary antibody that is used for the detection of the analyte that is present in the samples. Bridging ELISA, coming to bridging ELISA, it is a type of sandwich ELISA only, but it is most commonly used for the quantification of therapeutic drugs and the detection of anti-drug antibodies that are generated as an immunogenic response of the therapeutic drug. The anti-drug antibody forms a complex between the drug, and hence the name is called the bridging ELISA. The competitive ELISA, it's an inhibition or also known as blocking ELISA, where the analyte or the antibody competes for the similar antigen that is present in the sample to the known antigen which is present with the known antigen that is coated on the plate. Like RIA, the signal is inversely proportional in this case also. When the neutralizing antibody or competing antigen is present in the sample, the signal is lower. So when the antibody or the antigen is not available into the sample, the signal is higher. Coming to MSD, it's an advanced technology of an advanced form of ELISA, which uses the electrochemiluminescence for the detection of analyte in the samples. This is highly sensitive and advanced in the form of ELISAs. Ligand binding assays are versatile and widely accepted. They are used for the PK and TK studies, immunogenicity studies, biomarker analysis, receptor occupancy assays, and biotherapeutic developments. All these details we will see in the further slides. PK or pharmacokinetics. This is required for the dose escalation and dose finding studies, where we can identify the maximum tolerated dose and the safety related concerns that are associated with the dose of the drug. So it ensures the therapeutic efficacy as well as it minimizes the potential adverse effects. Also, it is dependent on the patient related factors as well as the drug related biochemical properties. Like patient related factors like genetic background, immune status, and the age of the patient or the age of the person also impact the PK profiles, as well as the drug related properties like dosing schedule, the amount of dose or the route of administration of the dose also impacts the PK profile of the drug. The drug may exist in different forms and is used like free drug, drug with one arm bound to the target, and the drug with both the arms bound to the target. All these forms of drug can be present in a body, and PK is used to quantify all these drug profiles or the drug concentrations at the site of action to understand the drug effect in the body. LBA can be designed to detect this free drug as well as the bound drug. So when coming to the detection of total drugs, the design of the assay or the assay formats that we select and the optimization of the reagents plays an important role. For total antibody detection or the total drug detection, the bridging ELISA format or the sandwich ELISA format is generally used. In this bridging format, the antibody pairs that detects the FC region of human IgG are usually used to detect the all forms of drugs, whether it is a free form of drug or it is a bound form of drug. The antigen that is coated, the antibody that is coated onto the plate bonds to the FC region of the antibody. Either it is free or conjugated, and the detection system also is against the FC region of the antibody, so that it can form a bridge between the antibodies, which is either free in form or bound in form. Apart from this, we can also use another way in which we can saturate the antibody with excess amount of antigens, so that all the sites available on the antibody are blocked and are in same form. So it becomes a completely bound antibody, and this completely bound antibody form can be detected by using antibodies which are against the antigens. So all these forms can be easily detected in one go. For detection of free and drug, we can go for indirect ELISA or sandwich ELISA or bridging format, where we can use the target or the anti-drug antibodies coated onto the plate and detecting the drug with the use of conjugated anti-FC antibody. So the anti-FC conjugated antibody will bind to the FC region of the drug, whether it is in the free form or it's a partially bound form. So in both the forms, it will detect the FC region of this antibody and it can quantify the amount of antigen that is present in free forms. Coming to next application, immunogenicity. Immunogenicity is required for accessing the potential of the drug to provoke an immune response. Whether the drug is having a potential to induce an immune response or not, we can detect by using a immunogenicity assay. The therapeutics are usually designed in such a way that they are non-immunogenic in nature and the immunogenicity is less. So thereby, if immunogenicity is high, it decreases the drug efficacy. And the immunogenicity assays are performed in 3 tiers. Tier 1 is a screening assay, which is a binding assay, which just shows the presence of ADA, whether the ADA is present or not in the samples. The next is confirmatory tier, where it is a inhibition ELISA, which confirms whether the antibody is whether the antibody is inhibitory in nature or not, or whether the ADA that is produced is against the target uh, target drug or not. So it gives a confirmation whether the antibody that has been produced is against the drug. And third tier is titration assay, where we get the magnitude of the ADA that is produced in the samples. Immunogenicity, as per FDA definition, immunogenicity is defined as the propensity of a therapeutic protein product to generate an immune response to itself and to related product, or to induce an immunologically related adverse clinical events. ADS can be of two types. Either it can be of neutralizing or it can be of non-neutralizing in nature, depending on the site of binding on the drug. If it is binding on the active site of the drug, they are neutralizing in nature. If they are not binding on the active site or anti-idiotypic sites, then they are non-neutralizing in nature. Non-neutralizing antibodies have less effect on the drug efficacy. Non-neutralizing antibodies binds to the therapeutic drug and they eliminates the drug through different mechanisms like ADCC, CDC, and ADCP. And the bridging ELISA is the common format that is used uh, for detection of ADS in the biological samples. Neutralizing antibodies plays an important role because they neutralize the drug and they eliminate the biological activity of the drug. When neutralizing antibodies are present in the sample, they bind to the active sites of the drug and they don't allow the drug to bind to the target site or the target receptors. Thus, they impact the patient safety and mediates a loss of drug efficacy by blocking the biological activity of the therapeutic drugs. There are two types of NAB assays. One is cell-based assay and the other is non-cell-based assays. So for identifying or quantifying the non-neutralizing antibodies, usually the competitive inhibition ELISA is used. If neutralizing antibodies are present in the drug in the samples, then they neutralizes the drug and the drug is not allowed to bound to the target or antigen that is coated onto the plate, thereby decreasing the or inhibiting the signal of the assay. If NAB is not present in the samples, then the drug goes and binds to the target and gives a positive signal. Thus, the signal of this assay is directly proportional to the amount of neutralizing antibodies that is present in the samples. If neutralizing antibodies are there, the signals will be less. And if the neutralizing antibodies are not there in the samples, then the signal will be high in the assays. The next application is biomarker analysis, which is required for identifying and quantifying the biomarkers that indicates the disease states or the drug response. Different biomarkers are generated or elevated based on the drug response or the disease state. The LBAs are used to identify and quantify these biomarkers. Usually the sandwich ELISA is used for the quantification of biomarkers that is present in the samples. There are different biomarkers that are available that are available, maybe prognostic, diagnostic, predictive, pharmacodynamic, safety, recurrence. And among these, we are mostly working on the pharmacodynamic biomarkers analysis. Now coming to the challenges that are associated with LBA formats. Specificity, sensitivity, interference, reagent quality, drug tolerance, and dynamic range. These are the general or few of the most common challenges that are faced during the development of LBSs. So whether it is a immunogenicity assay, PK assay, or NAB assays, these are the general challenges that are faced on a day-to-day basis. Next slide. Specificity and sensitivity. This is the most common challenge that is faced during the ADA and PK assay developments and validations. As per US FDA, for immunogenicity assays, the minimum required sensitivity should be 100 nanogram per ml, whereas for PK assays, it depends on the Cmax concentration of the drug. So at least the sensitivity of the assay should be 5% of the Cmax value that is reported for the biosimilars or the novel molecules. The sensitivity and specificity can be increased by selecting a specific formats or the reagents that are used for the method development. So there are few things that we can work on to increase the sensitivity of the assays. First is the selection of high affinity controls. As I previously also discussed, that high affinity controls plays a major role in the assay sensitivity. And as these LBAs are having a multiple washing steps, so there is a chance that the low affinity antibodies can be washed away, thus impacting or indirectly impacting the sensitivity of the assay. So the assay requires a high affinity positive control or the controls that can bind tightly to the drug or target so that they don't get washed away and the sensitivity or the required sensitivity of the assay can be achieved. So here is an example where we have worked on the immunogenicity assay based on bridging assay format, where we have used two different methods. One is sequential method and the other was homogeneous method for processing the samples. In the first sequential method, the positive control or ADA drug was added, anti-drug antibody was incubated along with the drug for certain time period and it was allowed to bind with the drug. And then the detection antibody was added. When this anti-drug antibody was added to the plate and allowed to bind with the drug that was coated on the plate, most of the anti-drug antibodies got bound to the coated drug antibody, and there was no free arms that were available to bind with the bioattenuated drug that was used for the detection system. As a result, the assay was unable to detect the anti-drug antibodies, and the sensitivity of the assay was low. So to overcome this challenge, we have changed the format to bridging format, where we have followed the homogeneous method, where the anti-drug antibody and the bioattenuated antibody was added together to the plate. So that anti-drug antibody was having chance to bind with both the coated drug as well as the bioattenuated anti-drug antibody, which is used for the detection. Thus, it was able to form the bridge between these two, and the signal was high, thereby increasing the sensitivity of the assay. As you can see in this sequential method, the sensitivity of the assay was 300 nanogram per ml, whereas in homogeneous method, when the sample was processed properly, the sensitivity of the assay came down to 39 nanogram per ml. Thus, there is a tenfold increase in the assay sensitivity. The other formats that we can use to increase the assay sensitivity is optimization of assay conditions. Assay condition also has an impact on the sensitivity of the assay. Like the antigen antibody interactions are higher at 37 degrees centigrade when compared to the reactions that are happening at 25 degrees centigrade. So by incubating the samples or the antigen antibody into 37 degrees centigrade can increase the assay sensitivity. The third is use of appropriate blocking agents. Blocking agents plays a major role in the background of the assay. So whenever the background is increased, and if the assay is based on the signal to noise ratio, then the signal to noise will decrease with the increase in the background noise, thereby indirectly impacting the assay sensitivity. Fourth is the use of signal amplification techniques. If the amplification is not proper, or if the signal is low, then we can improve this signal by by replacing the substrate by different luminescence or chemiluminescence substrate, or by using the ECL techniques, which are more sensitive and which are more sensitive in nature. Here is another example of sensitivity in PK assay, where we have seen that the sensitivity of the assay was impacting the selectivity parameter. As you can see here, the 10 different lots of samples or individual knife serums were used in the selectivity assay. All the samples were passing at HQC level, but at the LL level, there was an interference, or you can say that the sensitivity is low, and the recovery of these samples at the LL level was high. So to overcome this challenge, we have increased the assay sensitivity by reducing the coating concentration. Previously, the coating concentration that was used was 1 microgram per ml, and it was then reduced to 0.5 microgram per ml, so that the sensitivity of the assay can be increased. By increasing the assay sensitivity, we have seen that the selectivity samples which were failing previously at higher percent recoveries were found within the acceptance criteria of 80 to 120%. Next, coming to specificity. Specificity is an important parameter for LBS formats, because the assay needs to be very specific to the analyte of interest. They should be reacting only with the analyte of interest, but not with the cross-reacting molecules or the interfering molecules that are present in the assay. So to here is an example where we have seen the combinational therapy, where 3 different drugs were used for the same target or against the same target. In this is an example of PKSM, where we were supposed to detect only the analyte of interest, but as all these 3 molecules were having the same background, or that is human IgG background, FC region, the detection system that was used was against the anti-human IgG FC region. So as all these 3 molecules are having the same FC region, they were binding to the target as well as they are binding to the HRP or conjugate that was used in the assay. So all these 3 components were getting detected in the assay, and there was a false positive response because of this non-specificity. And to overcome this challenge, we have used the anti-idiotypic or anti-drug conjugate to detect the drug of interest. As you can see in the right hand side, the anti-drug antibody against the particular drug was used to detect the particular drug, whereas the other drugs which are interfering or which are similar to the drug were not able to detect, and the specificity was increased in this assay. Next slide. Target interference. This is a common challenge that is associated with the PD analysis. As you know that biomarkers are always present in the NAF serum, and this endogenous content of the biomarker always poses a challenge in the estimation of biomarkers in the samples. So whenever the endogenous content is present in the sample, this interferes with the analysis and they gives a false positive response or a higher concentration than the actual concentration of the biomarker that is present in the samples. So to remove this endogenous content of the samples, either we can go with a precipitation reaction or with a extraction method, where you can deplete this endogenous content and we can quantify the biomarker of our interest. The second method is we can we can introduce a MRD. We can dilute the samples so that the interfering molecules get diluted and the actual concentration of the analyte can be detected in the samples. The third approach is if you are unable to remove or deplete the endogenous content from the samples, or even if after applying the MRD or dilutions, even if the endogenous content is interfering with the sample analysis, then we can go with a statistical approach, which is either additional method or a subtraction method to check the analyte recovery. So the percent recovery is given by a formula, observed concentration or measured concentration by the actual or the nominal concentration of the analyte into 100. So in addition method, the expected or the nominal concentration was corrected by adding the endogenous content which is present in the samples. So the nominal concentration plus endogenous concentrations gives the expected concentration, and it is measured against the measured or actual concentration, which is obtained concentration from the analysis. In subtraction method, the net or the actual concentration that was measured was subtracted from the endogenous content. So the endogenous content is removed from the measured content, and the recovery is calculated by taking the ratio with the nominal concentration of the analyte that was spiked to the sample. Drug tolerance. This is a major issue associated with the ADF formats or immunogenicity formats. Drug tolerance is the sensitivity of the assay in presence of the known concentration of the drug. So whenever the drug is present, it always binds with the anti-drug antibody and it forms a complex, and hence interfering the uh, analysis or detection of the anti-drug antibody that is present in the samples. So it becomes a most challenging task to remove this drug or to dissociate the drug from the ADA and quantify the ADA that is present in the sample. So what should be the minimum drug tolerance or the maximum drug tolerance of the assay? So it always depends on the PK value of the drug, that is the Cmax value and the Ctrap value of the drug that is present in the sample. If the drug tolerance is equal to the Cmax value of the drug, then it is the ideal drug tolerance of the assay. If you are not able to achieve the Cmax value or of the drug, then the Ctrap should be the minimum drug tolerance that should be achieved in our assay. If you're failing to achieve the C-trough level of drug tolerance also in the assay, then we have to go for several different sample treatments where we can deplete the drug or we can remove the drug from the assay and we can achieve a higher drug tolerance in the assay. There are several sample treatments like ACE, BEAD, SPEAD, and P&A sample treatment techniques that can be used to increase the drug tolerance of the assay. And other simple techniques are acid dissociation, where you can use the acids with different strength to dissociate the drug from the ADA complex. And the other way is we can use the MRD optimization, where you can dilute the sample so that the drug concentration also gets reduced into the dilution. And we can quantify the or detect the ADA present in the sample. Fourth is the optimization of assay reagents. We can also work on the biotinylated reagent or the detection reagent, either by increasing or decreasing the concentration so that it can detect the ADA which are in minimum concentration also, so that the drug tolerance of the assay can be increased. The last and The last and most important is dynamic range. The LBA formats always face a challenge with the dynamic ranges. When you're going with the absorbance technique, the dynamic range is only 1 to 2 logs. So to increase this dynamic range, either we can go with chemiluminescence formats or electrochemiluminescence formats, where the dynamic range ranges from the 3 to 5 logs. The roles of LBS in clinical safety and efficacy, as we have already discussed, these LBSs are helpful to detect and quantify the anti-drug antibodies that are present in the samples, which can impact the drug efficacy, safety, and tolerability. Next, it plays a vital role in assessing the immunogenicity potential of the therapeutic drugs, whether they are immunogenic in nature or non-immunogenic in nature, thus helping to identify the potential risks early in the developmental process so that we can stop the clinical trials in the early stages or in the initial phases of the clinical trials. This also helps to ensure the patient safety throughout the clinical trials and post-market surveillance. As we have seen, this LBA are versatile in nature and widely accepted, and so they are reproducible and reliable techniques. This can be adapted to various applications, as we have seen previously that these are applied for PK, TK, immunogenicity, and biomarker assays. These are also accurate and sensitive. We can increase the sensitivity of the assays based on the formats that we are selecting, based on the positive controls that are used in the assays, and based on the optimization of the assay conditions. We can optimize the sensitivity of the assays, and these are most reliable and accurate assays. So they provide accurate and reliable quantification of the drug concentrations and ADA levels that are present in the samples. These are also widely accepted as these methods are most reliable, and especially for large molecules, these are well established and accepted across the global. These are relative samples as the LBS are based on the antigen antibody interactions only. So these are relatively straightforward to develop and validate compared to the other analytical methods that are much complex and complicated than these LBA formats. And these are few of the regulatory documents that are used for the validation and sample analysis of ligand binding assays. For immunogenicity assessments, immunogenicity testing of therapeutic protein products, that is by FDA, is mostly used for the development, validation, and sample analysis of anti-drug antibodies. ICH M10 guidelines on bioanalytical method validation and sample analysis are mostly used for PK analysis, which is published by ICH. And for PD analysis, we usually follow the FDA guidelines and Critical Path Institute's guidelines that's published by Critical Pathway Institutes. Now I hand over to Bhavika.
Thank you, Dr. Swaroop, for such an insightful session. We move to the next round, the question and answer Q&A sessions. My first question is, what should be the ideal background value of ligand binding assays and how to reduce the background of the assay?
Ideal background of the ligand binding assay. Usually the background of the ligand binding assays depends on the formats that we select, whether it is a non-competitive ELISA or it's a competitive ELISA, whether it is a absorbance based or a luminescence based assay. For non-competitive ELISAs, usually the background should be 1/3 of the LLOQ. And for competitive ELISAs, usually the S/N ratios are used for reporting. So the S/N should be at least 1.11 times of the lowest calibrator. For ADA that are non-quantitative assays, or if it is based on the absorbance, then the absorbance of less than or equal to 0.200 is acceptable. And for luminescence or RLU based assays, the RLU is less than or equal to 200 is acceptable as a background. And to lower the background, we can work on the blockers, we can work on the washing steps. Also, we can work on the conjugated antibodies that are used in the assay development. So we can use a blocking solution that can bind or that can block the plate efficiently and reduce the background or non-specific binding of the positive controls, thereby giving a higher background noise. And we can use lower washing steps, or we can use the detergents in the washing steps, which can remove the non-specific binding of the ligands. And third is the conjugated antibodies. We can decrease the concentrations of this ligand conjugated antibodies so that the signal or the background of the assay can be lowered.
Okay, okay. Thank you for that. Can we dilute the substrate for LBAs to reduce the intensity of the signal?
Dilute the substrate for ligand binding assays. No, usually it is not recommended because the reagents are been optimized in terms of the pH and the buffer concentrations. So usually it is not recommended to dilute the substrate to decrease or reduce the intensity of the signal. If you have to reduce the intensity of the signal, then we can work on the reagents like as I told you before, the enzyme linked conjugates. We can work on the dilution of these enzyme linked conjugates so that the intensity of the signals can be reduced.
Okay. Um, if an enzyme protein is already bound to ligand, what happens if we introduce another ligand that has higher affinity than the one in complex?
This we have already discussed in competitive ELISA, where the ligand which is having a higher affinity has a tendency or potential to remove the lower affinity antigen. So it is possible that it can replace the lower affinity antibody and the higher affinity antibody will form the complex with the antibody.
Okay. Um, my another question to you is it mandatory to include dilution QCs in our sample analysis runs?
This is a debatable query actually, because there are different practices followed by different industries. Some industries follows to include the dilution QCs and some industries follows not to include the dilution QCs. If I say as per ICH M10, dilution QCs are recommended only for the small molecules, but not for large molecules. So it depends on the institutes or the industries that they are practicing. Some industries practice to keep this dilution QCs as an indicator of sample dilution that they are doing so that they can judge whether the samples have been properly diluted or not. And some institutes sees as just a dilution of the QCs and they don't correlate with the dilution of the samples. So it's an industrial practice that is followed. And as per some papers of apps, they recommended that the dilution QC should be performed during the validations only, but not for the sample analysis. So I think it is not mandatory to include the dilution QCs in our sample analysis runs.
Okay, okay. Uh, I would like to take a pause here. Thank you, Dr. Swaroop, for answering all the questions. We understand that this session has kept us all very much engaged, but due to certain time constraints, we won't be able to take any further questions. But please be rest assured, we will be answering all your questions via email. And also, we would be sending a link of the recorded webinar to all your registered email IDs. It's been a real pleasure having you all us today. A heartfelt thank you to Dr. Swaroop Sarkar for such an insightful and thought-provoking session. On behalf of Veeda Lifesciences, thank you everyone for taking out the time and being part of this session. Your participation has made this session truly engaging. We hope you found the discussion valuable and we can't wait to connect with you again soon. Until then, stay well, take care. Thank you.
Thank you. Thank you, everyone.